Okay, let's unpack this. Have you ever looked at a living cell, or maybe just thought about the incredible complexity of, say, a plant or an animal, and wondered how it all works? It feels almost magical, right? Like there's some kind of vital force, maybe, something special that sets life apart from just ordinary chemistry.
That's a really great starting point because for centuries, yeah, that vital force idea, sometimes called animus, it really did hold sway. People
Got it.
thought there had to be something extra. But what's so fascinating now is how far our understanding has come. We know that living organisms, I mean, for all their incredible diversity and what looks like purpose, they are fundamentally chemical systems. Really sophisticated ones, mind you.
But chemical systems nonetheless.
Exactly. They don't break any laws of chemistry or physics, not at all. Instead, they leverage those laws in these extraordinarily intricate and honestly elegant ways.
And that's kind of our mission today, isn't it? To really take a deep dive into the foundational chemistry of life, pulling directly from the pages of molecular biology of the cell.
Hmm.
We're talking about, how cells are built, how they manage energy, how every single process from the tiniest bond formation up to the huge metabolic pathways, it's all interconnected and precisely controlled. Think of this as maybe your shortcut to understanding the real nuts and bolts of what makes life, well, alive.
Exactly. We'll dig into the core chemical components, look at the really remarkable properties of these giant molecules, macromolecules, how cells actually harness energy, and these incredibly organized metabolic networks that, keep everything going. It's truly about appreciating the elegance, the sheer ingenuity of cellular chemistry.
Okay, so if you're ready to get incredibly well informed about the chemistry view, let's jump right in. So let's start right at the very beginning. When we talk about the chemistry of life, what makes it unique compared to, say, the chemistry happening in a test tube or, I don't know, a rock?
Well, it mainly boils down to a couple of key things. First, life's chemistry overwhelmingly happens in water, in an aqueous solution. And second, it's built almost entirely on carbon compounds. Organic chemistry is really the chemistry of life.
Carbon.
Yeah, it's pretty astonishing when you think about it. Out of, 92 naturally occurring elements, life constructs itself predominantly from just 4. Carbon, hydrogen, oxygen, and nitrogen. CHON. I mean, those 4 make up something like 96, 97% of our body weight. And then there are a few others like phosphorus and sulfur that are crucial too, plus some trace elements.
Yeah.
But it's this relatively small palette of elements combined with carbon's amazing ability to form stable bonds with itself and other elements. That's the secret to life's incredible chemical diversity.
It's like nature decided to become a master chef with a very, very limited pantry, but learned to do amazing things with it. Okay, so you have these elements. How do they actually stick together to form molecules to build anything?
Right, that's where chemical bonds are essential. The really strong, sort of permanent connections that build the core structure of molecules are called covalent bonds. You can think of them like atoms sharing electrons in their outer shells. It forms a super strong, stable handshake, if you like.
Okay, handshake.
Yeah. And these bonds are incredibly robust. They resist the constant jostling and bumping from thermal energy at body temperature. They typically only break during specific biologically catalyzed reactions, and they don't just hold atoms together, they define the molecule's geometry, the specific angles and lengths between atoms. This stability is absolutely essential for building the long-lasting structures cells need.
Okay, so covalent bonds are the heavy-duty connectors, the structural beams.
Mm-hmm.
But what about the subtler interactions? I imagine cells need lots of delicate temporary connections too, right? For things to happen dynamically.
Precisely. You've hit on something crucial. That's where non-covalent bonds come into play. They're much, much weaker individually than covalent bonds, maybe 100 times weaker or even more.
Much weaker.
Yeah, but they are absolutely critical for molecules to recognize each other and interact dynamically, to associate and dissociate easily. Think of these non-covalent bonds like, I don't know, tiny little Velcro patches or weak magnets.
Okay.
Weak on their own, right? But imagine millions of them forming between 2 large molecules whose surfaces fit together perfectly like a hand in a glove. Collectively, they create an incredibly strong and specific attraction. It's strong enough to hold things together, but weak enough that they can come apart again when needed.
So that allows for things like enzymes binding to their targets or antibodies grabbing onto viruses.
Exactly. It allows proteins to fold into their precise shapes and then recognize and bind to exactly the right partners in the incredibly crowded environment of the cell. It's this delicate balance of many weak forces that gives life its amazing specificity and adaptability.
So these non-covalent bonds are the reason proteins can find the right molecule to interact with, or how the two strands of DNA can zip up and unzip. But all this intricate chemistry, it happens in a very specific environment, doesn't it?
Yeah.
And that environment is overwhelmingly.
Yep, H0. It makes up about 70% of a cell's weight, so virtually all the reactions of life happen in an aqueous environment. And water structure is the key to everything.
How so?
Well, a water molecule has covalent bonds between oxygen and hydrogen, but they're highly polar covalent bonds. Oxygen is more electronegative, meaning it pulls the shared electrons closer to itself. This gives the oxygen atom a slight negative charge and leaves the hydrogen atoms with slight positive charges. So the molecule acts like a tiny dipole, a little magnet.
Ah, okay, so that's where hydrogen bonds come in.
Exactly. These partially charged parts allow water molecules to form weak electrical attractions with each other and with other polar molecules. These are called hydrogen bonds. They're constantly breaking and reforming due to the thermal motion of the molecules, but collectively, these transient bonds give water its unique properties, why it's liquid at room temperature, why it has a high boiling point, high surface tension.
Right.
And these hydrogen bonds aren't just for water-water interactions. They are absolutely central to biology, forming between electronegative atoms like oxygen or nitrogen and an electropositive hydrogen atom attached to another electronegative atom. They're like invisible guide wires holding so many biological molecules together. Think protein structures, the DNA double helix. Fundamental.
It's almost like water itself is performing this constant, subtle dance that enables everything else to happen around it. And I know this leads to the whole concept of water-loving and water-fearing molecules, which is pretty fundamental, especially for how cells build membranes.
Absolutely essential. Molecules that have polar bonds or carry charges like sugars or DNA or most proteins are hydrophilic, literally water-loving. Water molecules are attracted to them, surround them, and readily dissolve them.
Okay.
On the flip side, you have molecules that are largely uncharged and nonpolar. They form few or no hydrogen bonds. Hydrocarbons, like the tails of fat molecules, are a prime example. These are hydrophobic or water-fearing. They don't dissolve well in water because forcing them into the water network disrupts the water's own hydrogen bonding. Water molecules essentially push them out of the way, and this property is ingeniously exploited in cell membranes.
How so? The membranes.
Well, cell membranes are built from lipid molecules that have a hydrophilic head and a hydrophobic tail. In water, they spontaneously arrange themselves into a bilayer, with the hydrophobic tails tucked away inside, avoiding water, and the hydrophilic heads facing outwards, interacting with the water inside and outside the cell. This creates a stable barrier.
A self-assembling barrier, just based on how the molecules interact with water. That's clever. So it's not just the strength of bonds, but how they interact with water that determines so much. And speaking of interactions, maybe we should quickly recap the main types of those non-covalent attractions that allow for all this molecular recognition. You mentioned hydrogen bonds.
Right, good idea. So besides hydrogen bonds, we have electrostatic attractions. These are basically attractions between charged groups. They're strongest between fully charged ions, what you might call ionic bonds, intercrystal, but they also occur between molecules with partial charges, those polar molecules we talked about. They're very important for specificity, though they are weakened a bit by water screening the charges.
Okay, electrostatic. What else?
Then there are van der Waals attractions. These are super weak, very short-range attractions that occur between any two atoms that are close together. They arise from temporary, fluctuating dipoles caused by the movement of electrons. Individually, they're tiny, but they become significant when many atoms on the surfaces of two large molecules fit together very closely, like puzzle pieces.
Like a final snug fit force.
Sort of, yeah. And finally, we have the hydrophobic force. Now, this isn't really a bond in the traditional sense. It's more of an effect driven by water. Water molecules prefer to hydrogen bond with each other, so they tend to push nonpolar surfaces together, minimizing the disruption to the water network. This force is incredibly important for driving things like protein folding, pushing the nonpolar amino acid side chains into the core, away from water.
So, okay, hydrogen bonds, electrostatic, van der Waals, and hydrophobic force.
Yeah.
Individually weak, but collectively, they add up to create highly specific and strong binding between molecules whose shapes are complementary.
That's the key takeaway. It allows for incredible selectivity in molecular interactions, which is the basis of almost everything a cell does.
It really paints a picture of a dynamic yet specific molecular dance. Now, another piece of this chemical stage setting is the concept of acids and bases. How do they fit in?
Right. So water itself can dissociate slightly, meaning an H0 molecule can release a proton, H+, which immediately associates with another water molecule to form a hydronium ion, HOA+. This happens constantly, with protons hopping rapidly between water molecules. An acid is basically any substance that increases the concentration of these HA+ ions when dissolved in water, usually by donating a proton. Strong acids, like hydrochloric acid, give up their protons easily. Weak acids, like the carboxyl group, COH, found in many biological molecules, hold onto their protons more tightly.
And bases do the opposite.
Exactly. A base is a molecule that decreases the HO+ concentration, usually by accepting a proton. Ammonia, or the amino group, NHers, are common weak bases in biology. Strong bases, like sodium hydroxide, NaOH, readily accept protons. The balance between acids and bases determines the pH of the solution, which is just a logarithmic measure of the HO+ concentration. Pure water has a pH of 7, which is neutral. The inside of a cell is kept very tightly regulated, close to this neutral pH, usually around 7.2 to 7.4, using buffer systems of weak acids and bases that can absorb excess protons or release them as needed.
So maintaining that precise pH is critical for everything to function correctly.
Absolutely critical. Even small shifts in pH can drastically alter the charge states of molecules, especially proteins like enzymes, and completely wreck their function.
It's incredible how tightly controlled everything has to be. Now, you mentioned carbon earlier as the backbone of life. Why carbon specifically? You hear about silicon-based life in sci-fi sometimes, but why did life here choose carbon? Silicon can also form 4 bonds.
Ah, yes, the silicon question. Carbon is truly exceptional. It has this unique ability to form 4 strong, stable covalent bonds, but critically, it can bond strongly to other carbon atoms, forming long chains, branch structures, and stable rings. Silicon can form chains, but the si-si bond isn't as stable, especially in the presence of water or oxygen. Carbon's versatility in forming complex, stable skeletons is just unmatched. It provides the perfect framework for the vast array of complex organic molecules needed for life.
So it's the stability and the versatility of the CC bond.
That's a huge part of it. And these carbon skeletons are then decorated with various chemical groups, specific combinations of atoms like the hydroxyl, OH, carboxyl, MagCOOH, carbonyl CO, phosphate, MagSPO, sulfhydryl, MagSH, or amino taxhero groups. Each of these groups has distinct chemical properties and reactivity, essentially giving different parts of the molecule a specific personality and dictating how it will interact and react.
So carbon provides a versatile skeleton, and these chemical groups are like the functional tools attached to it. Okay, so what are the main families of the small organic molecules, the building blocks that are constructed from this carbon scaffolding?
We can broadly categorize them into 4 major families. These are the small molecules, usually under 1,000 Daltons in mass, that serve as the basic units. First, we have sugars, or carbohydrates. Simple sugars like glucose are the primary fuel source for cells. They can also link up to form larger polysaccharides, like glycogen for energy storage or cellulose for structure.
Over sugars.
Second, fatty acids. These have a long hydrophobic hydrocarbon tail and a hydrophilic carboxyl group head. They're key components of cell membranes as phospholipids and are a major form of energy storage as fats or oils called triacylglycerols. We distinguish between saturated fats, no double bonds in the tail, and unsaturated fats with double bonds causing kinks.
Fatty acids, right, membranes and energy.
Third, nucleotides. These are really interesting. Each has 3 parts. A nitrogen-containing base, like A, G, C, T, or U, a 5-carbon sugar, either ribose or deoxyribose, and 1 or more phosphate groups. They are the monomers that build nucleic acids, DNA, and RNA, which store and transmit genetic information.
The information molecule.
Exactly. But nucleotides do more. ATP, adenosine triphosphate, is the main energy currency we talked about. Others act as signaling molecules within the cell, like cyclic AMP.
So very versatile. And the 4th family?
The 4th family is amino acids. There are 20 common types, each with a central carbon atom linked to an amino group, a carboxyl group, a hydrogen atom, and a unique side chain, or R group. These R groups vary widely in their properties. Some are small, some large, some charged, some hydrophobic. Amino acids are, of course, the building blocks of proteins.
Proteins, the workhorses. Okay, so these 4 families, sugars, fatty acids, nucleotides, amino acids, are the small monomers. That brings us naturally to the macromolecules. These are the big players, the giants of the cell, aren't they? Proteins, DNA, RNA, those complex polysaccharides. How do these smaller building blocks come together to form these cellular titans.
Right. Macromolecules are, by weight, the most abundant carbon-containing molecules in a cell. Proteins usually make up the largest fraction. And yes, most of them are polymers, which simply means they're long chains constructed by covalently linking together many of those smaller monomer subunits we just discussed.
Like beads on a string.
Pretty much, yeah. Sugars link to form polysaccharides, amino acids link to form proteins, also called polypeptides, and nucleotides link to form nucleic acids, DNA and RNA. Fatty acids are a bit different. They don't usually form huge polymers in the same way, but they assemble into large structures like membranes.
So how does the linking happen? If covalent bonds are strong, how do you make them?
The linking process typically happens through what's called a condensation reaction. In this reaction, one monomer provides a hydrogen atom, and the other provides a hydroxyl group, OH. When the bond forms between them, a molecule of water, HOO, is released or condensed out. Think of it like clicking Lego bricks together, and a tiny drop of water pops out each time.
Okay, water comes out. Does that require energy?
Yes, absolutely. Forming these covalent bonds to build polymers is energetically unfavorable. It requires an input of energy, like pushing a boulder uphill. The cell has to spend energy, usually derived from ATP hydrolysis, to drive these synthesis reactions forward.
And breaking them down.
Breaking them down is the reverse process, called hydrolysis, literally water splitting. A molecule of water is added across the bond, breaking it and releasing the individual monomers. This reaction is energetically favorable. It releases energy, like the boulder rolling back downhill.
So it's this constant cycle of condensation to build, requiring energy and hydrolysis to break down, releasing energy, that underpins how cells create and dismantle these massive structures like proteins and DNA. But these big molecules, they don't just exist as floppy chains, do they? Their specific three-dimensional shape is absolutely critical for what they do.
You've nailed it. That's perhaps the most amazing property of macromolecules, especially proteins and RNA. While the covalent bonds form the linear sequence, the primary structure, the chain doesn't just stay linear, it folds up. This folding is driven and stabilized by a multitude of those weak, non-covalent bonds we discussed earlier: hydrogen bonds, electrostatic attractions, van der Waals forces, and the hydrophobic effect forming within the same molecule. These forces guide the polymer chain to fold into a specific, stable, and unique three-dimensional shape called its conformation.
And that shape is everything.
Pretty much. This precise folding is vital because it creates unique contours, pockets, and surfaces on the macromolecule. These unique surfaces allow the macromolecule to bind with high specificity to other molecules, substrates for enzymes, signaling molecules for receptors, other proteins to form complexes. This selective binding, based on shape and chemistry complementarity, is the basis of nearly all cellular processes. It's how enzymes find their specific targets, how antibodies recognize antigens, how complex cellular machines like the ribosome assemble and function. It's truly an elegant system of molecular recognition driven by weak bonds arranging a polymer in 3D space.
Okay, this level of organization, this specificity driven by shape and weak bonds, it's mind-boggling. But it still feels like it's swimming upstream, thermodynamically speaking. It seems to fly right in the face of what I vaguely remember from physics class about this second law of thermodynamics. Doesn't that basically say everything in the universe tends towards greater disorder, towards chaos? My desk certainly agrees with that. But cells, they're creating intricate order. How do they get away with it?
Oh, the messy desk is a perfect, everyday example of entropy in action. And you're absolutely right to ask that. Cells do create incredible order. They build complex structures, they grow, they maintain themselves against decay. It looks like they're defying the second law. But the key is they aren't isolated systems. The second law applies strictly only to isolated systems, ones that don't exchange matter or energy with their surroundings. A living cell is an open system.
Okay, open system.
It continuously takes in energy from its environment, usually in the form of chemical energy from food molecules, or light energy from the sun for photosynthetic organisms. It uses some of this energy to create internal order, yes, but a significant portion of that energy is inevitably converted into heat during the chemical reactions. And this heat is released into the cell's surroundings. Heat is essentially disordered energy, the random movement of molecules. So by releasing heat, the cell increases the disorder, the entropy of its environment.
So they're basically exporting disorder.
Exactly. The increase in the disorder of the surroundings is always greater than the decrease in disorder, the increase in order within the cell itself. So if you consider the cell plus its surroundings, the whole system, the total entropy, the total disorder, always increases. The second law is perfectly satisfied. Cells are like little islands of order in a sea of increasing universal chaos, and they pay for their order by constantly releasing heat.
So they're basically paying an entropy tax to the universe to maintain their internal organization. Okay, that makes sense. And the first law of thermodynamics, that's about energy conservation, right? Energy isn't created or destroyed.
That's the one. Energy can be converted from one form to another: chemical energy to heat, light energy to chemical energy to kinetic energy for movement, but the total amount of energy remains constant. So, the chemical bond energy stored in the sugar molecules you eat isn't destroyed when your cells break them down. It's converted into other forms, like the heat that keeps your body warm, or the energy stored in ATP molecules, or the energy used to make your muscles contract. The crucial part for cells, connecting back to the second law, is what we call coupling. They have to link the energy-releasing reactions, which increase entropy, directly to the energy-requiring reactions, which create order. It's like a controlled burn rather than just letting the energy dissipate uselessly as heat. This coupling is how life actually harnesses energy to sustain itself.
Controlled burning. I like that. So how do cells actually get this energy from food? Is it literally like setting fire to sugar inside the cell?
Well, not quite as dramatic, thankfully. Although the overall chemical process combining fuel with oxygen to produce CoRO and water is similar to burning, cells do it in a very different way. They obtain energy by oxidizing organic molecules like sugars and fats. But crucially, it's a gradual, stepwise oxidation. It happens through a long series of controlled chemical reactions, each catalyzed by a specific enzyme. This allows the cell to capture the released energy in small, manageable packets, mainly by transferring it to those activated carrier molecules like ATP and NADH, rather than releasing it all at once as an explosion of heat.
Okay, so it's like dismantling a complex machine piece by piece to salvage the valuable parts instead of just blowing it up.
That's a great analogy. It maximizes the energy harvested in a useful form.
And this whole process of oxidation and energy extraction, it ties into the grand cycle of life on Earth, doesn't it? Plants making food, animals eating it.
Absolutely. It's a beautiful complementarity. Photosynthesis, carried out by plants, algae, and some bacteria, uses energy from sunlight to synthesize sugars and other organic molecules from CoRO and water. And as a byproduct, it releases oxygen into the atmosphere. Then, organisms like us perform aerobic respiration. We take in those organic molecules by eating plants or other animals and use the oxygen released by photosynthesis to break them down, extracting energy and releasing CoRO and water back into the environment. It forms this huge interconnected global carbon cycle. And similar biogeochemical cycles exist for other essential elements like nitrogen, phosphorus, and sulfur, constantly recycling the materials needed for life.
It's a planet-wide chemical engine. Okay, so the fundamental process behind that gradual breakdown, that gradual oxidation, is electron transfer, right? What we call oxidation and reduction. Can you break that down simply?
Sure. It sounds technical, but the core idea is straightforward. Oxidation, in a chemical sense, means the removal of electrons from an atom or molecule. Reduction is the opposite, the addition of electrons. A key thing to remember is that they always happen together. If one molecule loses electrons, is oxidized, another molecule must gain those electrons, be reduced. So we often talk about redox reactions.
Okay, one loses, one gains, like trading.
Exactly, and it doesn't have to be a complete transfer of an electron. Even shifts in the sharing of electrons within a covalent bond count. For instance, when a carbon atom bonds to a highly electronegative atom like oxygen, the oxygen pulls electrons away from the carbon, so we say the carbon has become partially oxidized. Conversely, when carbon bonds to hydrogen, carbon pulls electrons slightly towards itself, so it becomes partially reduced. A really useful rule of thumb in biology is to look at the number of CH bonds. An increase in CH bonds usually means reduction, while a decrease or an increase in CO bonds usually means oxidation.
That's helpful. More hydrogens, more reduced, more stored energy generally.
Generally speaking, yes. Reduced carbon compounds like fats and sugars store a lot of energy that can be released upon oxidation.
Okay, so cells have these pathways for gradual oxidation. But these chemical reactions, even the ones that release energy overall, they don't just happen spontaneously at body temperature, do they? Don't they need a little push to get started?
They absolutely do. Almost every chemical reaction, even a downhill one, requires an initial input of energy to get going. This is called the activation energy. It's like needing to push a car slightly uphill before it can start rolling down a bigger hill on its own. Molecules need to collide with enough energy and in the right orientation for bonds to break and reform. At body temperature, the random thermal energy often isn't enough to overcome this activation barrier for most biologically important reactions at a meaningful rate.
So how does life speed things up? This is where enzymes come in, right? The catalysts.
Exactly. Enzymes are the biological catalysts, and they are absolute masters at speeding up reactions. They are typically proteins, though some RNA molecules can also act as enzymes. What they do is lower that activation energy barrier for a specific reaction. They don't change the starting energy or the final energy of the reactants and products, just the height of the hill the reaction needs to climb to get started.
How do they physically do that? Lower the barrier.
They do it by binding specifically to the reactant molecules, which are called substrates. The enzyme has a unique pocket or groove called the active site that precisely fits the substrate, like a lock and key, or maybe more like a glove fitting a hand. By binding the substrate, the enzyme can hold them in the optimal orientation for the reaction, stress specific bonds, or directly participate chemically in the reaction mechanism. All of this stabilizes the transition state, that high energy intermediate point, effectively lowering the activation energy needed to reach it.
Wow. And they're specific.
Incredibly specific. Most enzymes catalyze only one particular reaction or a small set of very similar reactions. This specificity is crucial. It allows the cell to precisely control which reactions happen when and where, effectively steering molecules along specific metabolic pathways out of the thousands of possible reactions they could undergo.
So they're not just accelerators, they're also traffic directors for cellular chemistry, and they aren't used up in the reaction.
Nope. They emerge unchanged at the end and can go on to catalyze the same reaction over and over again, often thousands or even millions of times per second. They are incredibly efficient.
But wait, if they're speeding up reactions, does that mean they can somehow change whether a reaction is fundamentally favorable or unfavorable? Can they make an uphill reaction go downhill?
That's a really important point, and the answer is no. Enzymes cannot change the underlying thermodynamics, the equilibrium point of a reaction. They speed up both the forward and the reverse reactions equally. So if a reaction naturally favors the products, the enzyme helps it get there faster. If it naturally favors the reactants, the enzyme helps that equilibrium be reached faster. But it won't magically make more product appear than thermodynamics allows. They just accelerate the approach to equilibrium.
Okay, so they can't force energetically unfavorable reactions uphill. That brings us back to free energy, doesn't it? How does the cell actually make those necessary but uphill reactions happen, like building complex molecules?
Yes, this is where the concept of free energy G comes in. Free energy is a measure of the energy in a system that is available to do useful work. Chemical reactions involve a change in free energy, denoted as delta G. This Ag tells us whether a reaction can occur spontaneously under given conditions. If Ag is negative, the reaction releases free energy. It's considered energetically favorable or exergonic, and it can proceed spontaneously. These reactions tend to increase the disorder of the universe. If Ag is positive, the reaction requires an input of free energy to occur. It's energetically unfavorable, or endergonic. These reactions tend to create order and cannot happen on their own.
So how does the cell power those positive Ag reactions?
Through coupling. This is the biochemical accounting trick we mentioned. The cell links an energetically unfavorable reaction, positive A, to a separate, highly energetically favorable reaction, large negative Ag. As long as the overall Bg for the combined coupled process is negative, the whole sequence can proceed. It's like using the energy released by a falling rock, very negative D, to lift a bucket of water, positive D.
Okay, so it's about pairing reactions. Does the actual concentration of reactants and products matter for EG?
Yes, absolutely. The actual G depends not only on the intrinsic properties of the molecules, which is captured by something called a standard free energy change, AGG, but also very much on the concentrations of reactants and products. If you have a huge excess of reactants compared to products, that will tend to push the reaction forward, making the actual G more negative or less positive than the standard AG do. The cell often manipulates concentrations to help drive reactions.
Right, the law of mass action, basically. So the cell needs a reliable source of those highly favorable reactions to couple things too.
Yeah.
Where does that energy ultimately come from?
Primarily from the breakdown of food molecules, as we discussed. But the cell doesn't directly couple every biosynthetic step to food breakdown. Instead, it uses an intermediate strategy. It captures the energy released from food oxidation and stores it temporarily in a few types of special molecules called activated carrier molecules.
The energy shuttles.
Exactly. These molecules act like the cell's rechargeable batteries or delivery trucks. They pick up energy or chemical groups from energy-releasing reactions and transport it to where it's needed for energy-requiring reactions, like biosynthesis. They diffuse rapidly throughout the cell. They're often called coenzymes as well.
Okay, and the most famous of these, the absolute universal energy currency of the cell, has got to be ATP.
You got it. Adenosine triphosphate, ATP, it is far and away the most important and versatile activated carrier. It's formed by adding a third phosphate group onto adenosine diphosphate, ADP, a reaction that requires energy. Positive EG. This energy comes from coupling ATP synthesis to those highly favorable catabolic reactions, food breakdown.
So ATP stores energy. How does it release it?
It releases it when it's hydrolyzed, when one or sometimes two of its phosphate groups are split off by adding water. The hydrolysis of ATP to ADP and inorganic phosphate pi is a highly favorable reaction, releasing a good chunk of free energy, a large negative bond. This energy release happens partly because you're breaking a high-energy phosphoanhydride bond and relieving some repulsion between the adjacent negative charges on the phosphate groups, and also because the products, ADP and pi, are more stable.
So the cell spends ATP by breaking that bond.
Well, it's more often used in coupling. The energy from ATP hydrolysis is used to drive an unfavorable reaction, often by transferring that terminal phosphate group directly onto one of the reactants. This phosphorylation creates a high-energy intermediate, making the subsequent reaction step favorable.
So it activates the molecule first.
Precisely. For example, to join two molecules A and B together, which requires energy, the cell might first use ATP to transfer a phosphate to molecule A, forming A phosphate. This A phosphate is now activated, high energy, and can readily react with molecule B to form AB, releasing the phosphate. A to C provides the energy push via that intermediate step.
Clever. So ATP is the main energy currency.
Yeah.
But you mentioned other carriers, particularly for redox reactions.
Yes. Besides carrying energy in phosphate bonds, cells need to carry high-energy electrons. The main carriers for this are NADH and NADPH. These molecules, derived from the vitamin niacin, exist in an oxidized form, NADDO and NADP, and a reduced form, NADH and NADPH. The reduced forms carry two high-energy electrons plus a proton, effectively a hydride ion and a TPA.
Okay, electron taxis. Is there a difference between NADH and NADPH? They sound almost identical.
They are very similar, differing only by a single phosphate group located away from the electron-carrying part. But this small difference is crucial because it allows them to be recognized by different sets of enzymes. Generally, NADPH operates primarily in anabolic pathways, reactions that build up larger molecules from smaller ones. It provides the high-energy electrons, reducing power, needed for biosynthesis. Cells typically maintain a high ratio of NADPH to NADP. NADH, on the other hand, is primarily involved in catabolic pathways reactions that break down food molecules. It collects high-energy electrons from food oxidation and then delivers these electrons to the electron transport chain to ultimately generate ATP. Cells usually have a much higher ratio of NAU to NADH.
So that extra phosphate group acts like a label, directing NADPH to building reactions and NADH to energy generation.
That's a perfect way to think about it. It keeps the electron pools for catabolism and anabolism largely separate and independently regulated.
Fascinating separation of duties. Are there other important carrier molecules besides ATP, NADH, and NADPH?
Oh yes, quite a few, specialized for carrying other chemical groups. For example, acetyl-CoA carries an acetyl group, a 2-carbon unit, linked via a high-energy thioester bond. It's absolutely central in metabolism, delivering the acetyl group to the citric acid cycle and also using it for biosynthesis. FADH is another important electron carrier, similar to NADH, often involved in fat breakdown. Then you have carriers for carboxyl groups, like carboxylated biotin, methyl groups, S-adenosilmethionine, or SAM, glucose units, UDP glucose, and others. Each plays a specific role in transferring particular building blocks or energy packets.
It's like a whole toolkit of specialized delivery trucks within the cell. And many of them seem to involve nucleotides, like ATP or the A in CoA.
Yeah.
Any significance there?
That's a sharp observation. Yes, many coenzymes contain an adenosine diphosphate ADP portion. One hypothesis is that this might be a molecular fossil, a relic from an early stage of evolution, perhaps an RNA world where RNA molecules played more central catalytic and information-carrying roles, and nucleotides were readily available building blocks.
An echo of ancient life. Okay, so bringing this together. Energy from food is captured by activated carriers like ATP and NADH, NADPH. How is this energy then used to actually build the giant polymers of life proteins, nucleic acids, polysaccharides?
The synthesis of these biological polymers follows a common principle. It involves the repetitive addition of monomer subunits via those condensation reactions we talked about, releasing water. And since condensation is energetically unfavorable, each step requires energy input. This energy is usually provided by coupling the condensation step to the highly favorable hydrolysis of a nucleoside triphosphate, most often ATP, but sometimes GTP, CTP, or UTP, especially in nucleic acid synthesis. Typically, the monomer itself, or the growing end of the polymer chain, is first activated by linking it to a high-energy intermediate, often involving ATP, and then this activated intermediate reacts to add the next monomer, releasing the activating group in an energetically favorable step.
So it's always that theme of coupling an unfavorable joining reaction to a very favorable energy-releasing reaction, often involving those triphosphate carriers.
Exactly. That fundamental strategy underlies the construction of nearly all the complex macromolecules that make life possible.
All right, so we've got the building blocks, the energy currency, the enzymes, the principles of coupling. Let's now trace the journey of, say, a molecule of glucose from your breakfast cereal once it gets inside one of your cells. How does the cell actually start breaking it down to extract that energy?
Okay, so the first thing that happens, even before the glucose gets into the cell usually, is digestion. Large food molecules, complex carbohydrates like starch, proteins, fats, are broken down into their smaller monomer subunits. Simple sugars like glucose, amino acids, fatty acids, and glycerol. These smaller molecules can then be absorbed by cells. Once inside the cell's cytosol, these fuel molecules begin their journey of gradual oxidation to release their stored energy.
And for glucose, the starting point, the almost universal first stage, is glycolysis, right? You mentioned it doesn't need oxygen.
Precisely. Glycolysis, which literally means sugar splitting, is a sequence of 10 enzyme-catalyzed reactions that takes place in the cytosol of probably almost all living cells on Earth. And yes, crucially, it does not require molecular oxygen. This strongly suggests it's a very ancient metabolic pathway, likely evolving early in life's history when the Earth's atmosphere lacked significant oxygen.
10 steps.
Huh?
What's the bottom line? What goes in and what comes out?
What goes in is one molecule of the 6-carbon sugar, glucose. What comes out after those 10 steps are 2 molecules of the 3-carbon molecule called pyruvate. Along the way, there's a net production of energy carriers. Specifically, for each glucose molecule, glycolysis yields a net gain of 2 ATP molecules and 2 NADH molecules. It's important to note that 4 ATPs are actually produced, but 2 ATPs are consumed in the early investment steps, so the net is 2.
Okay, 2 ATP and 2 NADH plus the 2 pyruvate molecules. That doesn't sound like a huge energy yield from a whole glucose molecule.
It's not, compared to the total energy available in glucose. Glycolysis only extracts a small fraction, but it's quick, it doesn't need oxygen, and it provides the essential starting material, pyruvate, for further energy extraction if oxygen is available. And those 2 ATPs can be vital, especially under anaerobic conditions.
Right. So what happens if there's no oxygen available, say in our muscle cells during really intense exercise or for yeast making beer or bread? Glycolysis produces NADH, but the cell needs to get NAD plus back to keep glycolysis running.
Exactly. That's where fermentation comes in. In the absence of oxygen, the pyruvate and the NADH produced by glycolysis stay in the cytosol. To regenerate the NADH needed to keep glycolysis going, specifically for step 6, the cell needs to dump the electrons from NADH onto something else. In fermentation, pyruvate itself, or derivative of it, acts as the electron acceptor. For instance, in vigorously exercising muscle cells, pyruvate is reduced by NADH to form lactic acid. This regenerates NADO. In yeast, pyruvate is first converted to acetaldehyde, which is then reduced by NADH to form ethanol, and co-urate is released. Again, NADU is regenerated.
So fermentation is basically a way to keep glycolysis running, generating that small amount of ATP by finding a way to recycle NADH back to NAD+ without using oxygen.
Precisely. It sacrifices the pyruvate, which still contains a lot of energy, just to keep the ATP production from glycolysis going. It's much less efficient overall than complete oxidation, but it's a vital survival strategy when oxygen is scarce.
Okay, now cells are smart. They don't just burn all their fuel the moment they get it. They need ways to store energy for later. How do they do that?
Very true. Organisms need to maintain a high ATP-ADP ratio to power cellular processes, but food intake is often periodic. So energy storage is crucial. In animals, the primary form of long-term energy storage is fat. Fats, chemically known as triacylglycerols or triglycerides, are stored mainly in specialized cells called adipocytes, fat cells. Fats are incredibly efficient for storage. Why? Because they're hydrophobic, they don't associate with water, so you store almost pure fuel. And gram for gram, fats store about twice as much energy as carbohydrates like glycogen.
So more energy packed into less space and less weight.
Exactly. An average adult might have enough stored fat to survive for about a month without food. For shorter-term energy needs, animals store glycogen. This is a branched polymer of glucose units, stored mainly as small granules in the cytoplasm of liver and muscle cells. Glycogen reserves typically provide enough energy for about a day.
And what about plants?
Plants also store energy, of course. During photosynthesis, they produce sugars. They can store excess sugars short-term as starch, which is analogous to glycogen, another glucose polymer, just branched differently, often within their chloroplasts. For longer-term storage, especially in seeds, plants often store large amounts of both fats, oils, and starch. Think of nuts, corn kernels, soybeans packed with energy reserves for the developing embryo.
So fats and complex carbs are the main storage forms. Now let's get back to the breakdown. What happens to that pyruvate from glycolysis and the fatty acids from fat breakdown when oxygen is available? Where do they go next for more energy extraction?
When oxygen is present, these fuel molecules head into the mitochondria, which are often called the powerhouses of the eukaryotic cell. Fatty acids are imported directly into the mitochondrial matrix, the inner space. There, they undergo a process called beta-oxidation. This is a cycle of reactions that sequentially snips off 2 carbon units from the fatty acid chain in the form of acetyl-CoA. Each cycle also produces 1 molecule of NADH and 1 molecule of FADH, another electron carrier.
Okay, fatty acids become acetyl-CoA plus electron carriers inside the mitochondria. What about pyruvate?
Pyruvate, which was produced by glycolysis out in the cytosol, is actively transported into the mitochondrial matrix. Once inside, it encounters a large enzyme complex called the pyruvate dehydrogenase complex. This complex carries out a crucial conversion. It decarboxylates pyruvate, removes a co-ester molecule, oxidizes the remaining 2-carbon fragment, and attaches it to coenzyme A, forming acetyl-CoA. This reaction also produces 1 molecule of NADH per pyruvate.
So both sugars, via pyruvate, and fats ultimately get converted into this common 2-carbon unit, acetyl-CoA, inside the mitochondria. It seems like acetyl-CoA is a major hub.
It absolutely is. It's the main product derived from the breakdown of both carbohydrates and fats, and it holds most of the readily usable chemical energy captured from them up to this point. It's now poised to enter the next major stage of energy extraction.
And that next stage, the big one inside the mitochondria, is the legendary citric acid cycle, also known as the Krebs cycle or the tricarboxylic acid cycle, TCA cycle. This sounds really central.
It truly is central to aerobic metabolism. The citric acid cycle takes place entirely within the mitochondrial matrix in eukaryotic cells. It accounts for about 2/3 of the total oxidation of carbon compounds derived from food in most aerobic organisms. What happens is the 2-carbon acetyl group from acetyl-CoA enters the cycle by combining with a 4-carbon molecule called oxaloacetate. This forms a 6-carbon molecule, citrate, hence the name citric acid cycle.
Okay, 2 carbons in, joins 4 carbons, makes 6 carbons. Then what?
Then, through a series of 8 enzyme catalyzed steps, this citrate molecule is progressively oxidized. Along the way, carbons are released as carbon dioxide, CO, which is the waste product we exhale. High-energy electrons are harvested and transferred to electron carriers, producing NADH and FAD arrows. A small amount of energy is also captured directly as GTP, guanosine triphosphate, which is energetically equivalent to ATP and easily converted to it. And the crucial part is that the final step of the cycle regenerates the starting 4-carbon molecule, oxaloacetate, allowing the cycle to accept another molecule of acetyl-CoA and begin again. It's a true cycle.
So for each turn, starting with 1 acetyl-CoA, what's the harvest?
Per acetyl-CoA entering the cycle, 2 molecules of co-arrows are released, 3 molecules of NADH are generated, 1 molecule of FADH is generated, and 1 molecule of GTP or ATP is produced.
Wow, that's a significant haul of electron carriers. Does the cycle itself use oxygen directly?
No, the reactions of the citric acid cycle itself do not directly consume gaseous oxygen. However, it's considered part of aerobic respiration because it relies heavily on the next stage, which does require oxygen, to regenerate the NADU and FAD from the NADH and FAD arrows produced by the cycle. Without oxygen, the cycle would quickly grind to a halt due to lack of NADU and FAD.
Right, it needs the downstream process to keep its own input supplied. Okay, so the cycle has churned out loads of NADH and FADH, which are packed with high-energy electrons stripped from the original food molecules. This leads us to the final and most productive stage of energy harvesting, electron transport and oxidative phosphorylation. This is where the big ATP payoff happens.
This is absolutely where the vast majority of ATP from aerobic respiration is made. This process also occurs in the mitochondria, specifically involving protein complexes embedded in the inner mitochondrial membrane. The NADH and FADH are generated from glycolysis. The NADH needs to be shuttled in. Pyruvate oxidation and the citric acid cycle now donate their high-energy electrons to an electron transport chain within this membrane.
A chain of molecules passing electrons.
Essentially, yes. It's a series of protein complexes and small mobile electron carriers that sequentially accept and donate electrons, passing them down an energy staircase. Each transfer releases a small amount of energy. Critically, several of the protein complexes in the chain use the energy released during electron transfer to actively pump protons, halions, from the mitochondrial matrix across the inner membrane into the inner membrane space, the space between the inner and outer mitochondrial membranes.
Pumping protons out, creating a gradient.
Exactly. This pumping establishes a steep electrochemical proton gradient across the inner membrane, a difference in both proton concentration, pH, and electrical charge. This gradient represents a form of stored potential energy, like water stored behind a dam. It's often called the proton motive force, and the electrons, having cascaded down the chain, finally reach the end where they are transferred to molecular oxygen, which combines with protons from the matrix to form water. This is the only step in the entire process of glucose breakdown that directly consumes the oxygen we breathe.
So oxygen is the final electron acceptor, the end of the line for those electrons. And the proton gradient is the stored energy. How does that make ATP?
The energy stored in the proton gradient is then harnessed by a remarkable molecular machine also embedded in the inner mitochondrial membrane called ATP synthase. Protons flow back down their electrochemical gradient from the inner membrane space into the matrix, passing through a channel within ATP synthase. This flow of protons drives a rotor-like component of the enzyme, causing conformational changes that catalyze the synthesis of ATP from ADP and inorganic phosphate. Pi. It's like water flowing through a turbine to generate electricity. This process of ATP synthesis, driven by the energy released from electron transport, is called oxidative phosphorylation.
Oxidative, because it starts with electron donation, ultimately requiring oxygen, and phosphorylation, because it adds phosphate to ADP. That's amazing. A molecular turbine. And this makes way more ATP than glycolysis.
Oh, vastly more. While glycolysis yields a net of 2 ATP per glucose, the complete oxidation of glucose through the citric acid cycle and oxidative phosphorylation yields somewhere around 30 to 32 additional ATP molecules. It's much, much more efficient.
So the mitochondrion really is the central hub for energy conversion in aerobic cells, bringing together the breakdown products of sugars and fats, running the citric acid cycle, and then using electron transport and that proton gradient to make the bulk of the cell's ATP.
Absolutely. It integrates all these energy-yielding processes. But it's important to remember one more thing about these pathways, especially glycolysis and the citric acid cycle. They aren't just about breaking down molecules for energy catabolism.
Well, what else do they do?
They also serve as a crucial source of building blocks for making new molecules, anabolism. Many of the intermediate molecules generated during glycolysis and the citric acid cycle can be siphoned off from these pathways and used as precursors to synthesize amino acids, nucleotides, lipids, and other essential small organic molecules that the cell needs to grow and repair itself. For example, oxaloacetate and alpha-ketoglutarate from the citric acid cycle are starting points for synthesizing several amino acids. It's a beautifully integrated system where the same central pathways provide both energy and biosynthetic precursors.
So it's a two-way street, providing fuel and building materials simultaneously. That's efficient. And finally, just briefly, what about essential elements beyond CH and O, like nitrogen and sulfur? How do they fit into this grand metabolic scheme? Where do we get them?
Right, nitrogen is absolutely vital, mainly for building amino acids for proteins and nucleotides for dRNA. The ultimate source is atmospheric nitrogen gas, but most organisms can't use it directly. Certain microorganisms perform nitrogen fixation, converting NU into usable forms like ammonia. Animals, including us, typically get our nitrogen by eating proteins and nucleic acids in our diet. We can synthesize about half of the 20 standard amino acids ourselves, often using intermediates from glycolysis or the citric acid cycle as starting points. But the other half, the essential amino acids, we lack the pathways to make, so they must be obtained from our diet.
Got it.
And sulfur.
Sulfur is needed mainly for a couple of amino acids, methionine and cysteine, and some coenzymes. Again, plants and many microorganisms can take up inorganic sulfate from the environment and reduce it to the sulfide form needed for biosynthesis. Animals generally rely on obtaining sulfur-containing amino acids from their diet.
It really is just incredible to step back and consider the sheer scale and complexity of it all. Millions of reactions happening every second within just one tiny cell, all these pathways intricately interconnected, branching, converging. How does a cell possibly manage such an immense chemical traffic jam and maintain such precise balance?
That is perhaps the ultimate question, and the answer lies in regulation. A cell is indeed an incredibly intricate chemical machine. Despite the mind-boggling number of potential reactions and pathways, thousands have been mapped, a cell's metabolism is remarkably stable under normal conditions, yet also highly adaptable to changes like starvation or disease. This stability and adaptability depend on an elaborate network of control mechanisms that constantly monitor the cell's needs and adjust the rates of key metabolic reactions accordingly. This regulation often involves feedback inhibition, where the end product of a pathway inhibits an enzyme acting early in that pathway, preventing wasteful overproduction. It also involves sophisticated control over gene expression, ensuring the right enzymes are made at the right times. Ultimately, all this regulation relies on the exquisite ability of proteins, particularly enzymes, to change their shape and chemical activity in response to binding other molecules, sensing the cell's internal state and external environment. It's a dynamic, self-adjusting system of phenomenal complexity and elegance. Hashtag tag tag outro.
So, okay, wrapping this up,
Right.
what does this all really mean? We started off kind of questioning the magic of life, didn't we? And what we seem to have found is something, honestly, even more incredible. It's this world built entirely on understandable fundamental chemistry, where every single reaction, every molecule, every energy transfer is precisely, intricately orchestrated.
Yeah.
It's not magic. It's just astonishingly elegant chemical engineering.
Yeah, and if you connect that to the bigger picture, it really underscores that life isn't just some random jumble of reactions happening in a bag. It's a highly organized, self-regulating chemical system. It's constantly working, actively working against that universal tendency towards disorder, the second law, by meticulously converting energy and raw materials into the very fabric of its own existence. It truly is a testament to the power of molecular interactions and controlled energy flow.
Absolutely. From, the delicate dance of those non-covalent bonds folding a protein just right, to the incredibly efficient enzyme-driven pathways like glycolysis and the citric acid cycle that are literally powering you as you listen to this. Hopefully, you now have a much deeper appreciation for the chemical symphony playing out constantly in every single one of your cells. It really does make you appreciate that invisible world inside us.
And thinking forward, this deep understanding raises such important questions, doesn't it? Knowing that life operates fundamentally within these constraints of physics and chemistry, how can we best leverage this knowledge for developing new, more targeted therapies for diseases, for engineering microorganisms to produce useful chemicals, maybe even for designing synthetic life forms from the ground up? The basic principles we've talked about today, they really are the essential starting point for so much biological innovation.
It's a great point. It's foundational knowledge with huge potential. Well, hopefully this deep dive gives everyone listening a whole new appreciation for the hidden but absolutely vital chemical factories chugging away inside each of us. Thank you so much for joining us on this exploration of cellular chemistry. Keep learning, keep questioning, and until next time, stay curious.